inositol triphosphate, IP3, diacylglycerol cascade. So we have cells, right, and it is a cellular membrane, outside, inside, and this cell we have GPCR receptor, and as we have ligand, and this ligand attaches to this receptor, this causes conformational changes of this receptor. Yeah, inside the cell, this receptor coupled with so-called G protein, and this G protein has three domains: alpha, gamma, and beta domain. Alpha domain also bounded with GDP, guanosine diphosphate, and as we have binding of a ligand, it causes conformational changes, and it causes detachment of alpha G protein and substitution of GDP to GTP, guanosine triphosphate. Yeah, so it becomes phosphorylated, and as it becomes phosphorylated, it activates another enzyme that present in the cell membrane. It's called phospholipase C. And phospholipase C cleaves phosphatidyl inositol biphosphate, into two parts. The first part is a diacylglycerol, DEG, and another part is the inositol triphosphate. They have a little bit different properties, DEG and inositol triphosphate. DEG still hydrophobic, and it's still be present inside the bilipid layer of plasma membrane. And inositol triphosphate is a polar molecule, so it very well dissociates and very well diffuses, yeah, in the water. So what is the next? We have endoplasmic reticulum, and in this endoplasmic reticulum we have IP3 sensitive calcium channels. So inositol triphosphate binds to IP3 sensitive calcium channels, activate them, and causes releasing of calcium to intracellular space. Calcium ions bind to calmodulin, and produce calcium calmodulin complex. And this complex important for stimulation of different proteins, and different protein kinases. So we have stimulation of protein kinases, and one of these protein kinases is a protein kinase C. So we have stimulation of protein kinase C, to which diacylglycerol is bounded also. And as a result, we have cellular effect.